برای امتحان میاد. مطالب فیلم‌ها برای امتحان نیست. صفحه من رو می‌بینید؟ من یه فیلمی هست راجع به مسیر گلوتاتیون که مربوط به پنتوز فسفات هست. من اول این رو خدمتتون میذارم، بعد میرم سراغ اون یکی فیلمم.
Welcome back to Dirty Medicine's Dirty Biochemistry series. This video is part 2 of the mini-series, and what I mean by that is that this is the follow-up video to the video that you probably just watched on the HMP shunt, also known as the pentose phosphate pathway. As I alluded to in the beginning of that video, this is the second part of a biochemical process that is very closely tied to the HMP shunt. As a quick refresher, in that part 1 of the HMP shunt video, we discussed how NADPH is generated by the HMP shunt. We talked about the one step that you should know, how it's generating NADPH, and that NADPH would be very closely tied to what we're going to talk about in this video in part 2 about glutathione reductase. Let's jump right into part 2 and complete your understanding of not only everything that we talked about with the HMP shunt, but also transition to glutathione and free radicals. So in this video, we're going to be talking about glutathione reductase. This pathway has one goal, and it's to reduce glutathione. Now, sometimes this pathway is referred to as the glutathione pathway, other times it's referred to as the glutathione reductase pathway, but technically speaking, glutathione reductase is an enzyme. So it's a little silly to name a pathway after an enzyme. So if you hear me use the word glutathione reductase pathway or glutathione pathway or free radical pathway, I'm talking about the same thing. Okay? So this is the glutathione pathway, and its only goal is to reduce glutathione. Now, let me turn your attention to a slide that we had in the first video in the HMP shunt. We said that free radical detoxification requires reduced glutathione. So obviously, in this glutathione pathway, the reason that the goal is to reduce glutathione is to be able to process and detoxify free radicals and peroxides. Now, again, just to really complete the whole picture, it was free radical detoxification required reduced glutathione. And in this pathway, I'll show you how we actually reduce that glutathione. That required NADPH came from the HMP shunt. So that all of these are tied together. Now, what you need to know about the glutathione reductase pathway. So you start with oxidized glutathione, right? So you have glutathione, it's just existing in an oxidized form. But that's not good because the body, in order to make use of its normal physiologic biochemistry, needs it to be in its reduced form. So the enzyme that reduces oxidized glutathione to reduced glutathione is glutathione reductase. And just look at the name of the enzyme, glutathione reductase or reductase. So it's reducing glutathione. So it tells you that glutathione reductase is reducing glutathione. So it's once glutathione is in its reduced form that it can actually go ahead and carry out its true biochemical purpose. And that is to say that reduced glutathione will proceed to detox the body against free radicals and to detox the body against peroxide. Okay, so in this very simplified image, what you see is reduced glutathione positively converting peroxide into water, and also in green text, handling free radical detoxification. So glutathione is a very potent free radical detoxifier, and it's the reduced form of glutathione that we need in order to protect the body against free radicals. So if you were medical, probably free radicals are very serious, and it's when the body cannot process or detoxify those free radicals that cellular damage occurs. So again, if I can sort of take a step back and pause for just a second, what I'm telling you is that the glutathione pathway has one goal. It's to reduce glutathione, and that reduced glutathione will go on and chop up all the free radicals that could potentially cause really bad damage and disease in the human body. So that's the biochemistry. Again, that process of getting reduced glutathione depends on the HMP shunt. Now, let me tie this all together. In this step, where the enzyme glutathione reductase is happening, NADPH is actually being converted back into NADP+. And this is interesting, right? Because I told you that the goal of this pathway was to reduce glutathione. But we need NADPH to do that. So we need an input of NADPH in order to reduce glutathione. So where do we get that input of NADPH? Well, this is the connection between the HMP shunt and the glutathione pathway. We're taking the NADPH that was generated here in the HMP shunt, and we're plugging that NADPH into the glutathione pathway and connecting these wheels, if you will. So what you see is constant spinning or constant wheels, where these two pathways are interacting with one another in a very cyclical form. So what you're seeing on this slide is a very simplified version of the connection between the HMP shunt shown on the left and the glutathione reductase pathway shown on the right. To once again summarize, because I cannot stress enough how high yield this is, glucose 6-phosphate dehydrogenase will take glucose 6-phosphate, which it stole from glycolysis, and convert it into 6-phosphogluconolactone. The only reason that it's doing that is to spit out NADPH from NADP+. Then glutathione reductase sees that NADPH sitting there, and it goes, I need that NADPH in order to reduce glutathione. And reducing glutathione is of the utmost importance because, again, the body needs reduced glutathione in order to handle free radicals. So that the glutathione reductase will grab that NADPH that the HMP shunt generated, and it will use it to make reduced glutathione. And of course, when it makes reduced glutathione, one of the end products that the glutathione pathway spits out is an NADP+, which the HMP shunt looks at and says, okay, now I'm going to keep going. And you've got this cyclical wheel, if you will, that just keeps spinning. So this is the connection between the HMP shunt and the glutathione reductase pathway. Now, what happens if cells lack NADPH, right? That is to say, what happens if this wheel stops spinning because one of the inputs is no longer there? Well, I'm going to show you in purple what would happen. Let's say that you don't have NADPH, and for now, don't worry about why you don't have it. But if you don't have NADPH, then you get a decrease of reduced glutathione, because glutathione reductase can only reduce glutathione if it also converts NADPH to NADP+. So a decrease of NADPH means a decrease in reduced glutathione. And of course, a decrease in reduced glutathione means an increase in oxidative stress. And this is really serious. When free radicals go unchecked and there's increased oxidative damage in the body, disease will occur. So the question is, what causes oxidative stress? Well, we've got a few different causes. There are sulfa drugs, the drug primaquine, infections or any kind of illness, nitrofurantoin, another antibiotic, and fava beans. Kind of random, I know, but fava beans. So how do you remember the causes of oxidative stress? Well, what I want you to remember is spin-F, or spin-off. So instead of saying spin-off, I say spin-F, spin-F. And the reason that spin-F reminds me of these different causes of oxidative stress is that spinning is what a wheel does. And I remember the little wheel, or the cyclical nature of the HMP shunt and the glutathione reductase pathway. So again, how do you remember that this mnemonic correlates to oxidative stress? Well, I remember the spinning of the wheels in the HMP shunt and the glutathione pathway. Then I remember spin-off and spin-F, S for sulfa, P for primaquine, I for infection, N for nitrofurantoin, and F for fava beans. So these are the things that cause oxidative stress. And this is really significant because, as we're going to see in just a moment, in certain people who lack an enzyme, these insults that cause more oxidative stress that the body could normally handle just fine will actually go unchecked and cause so much oxidative stress that you're going to get really severe anemia. So let's come back to the question, what happens if cells lack NADPH? Well, let's talk about one major cause of that. So one way that cells can lack NADPH and be really susceptible to the agents that I just showed you on the last slide that cause significant oxidative stress is if you knock out glucose 6-phosphate dehydrogenase. So those who have a deficiency of glucose 6-phosphate dehydrogenase obviously cannot generate NADPH from NADP+. And this is a problem because when we can't generate NADPH, glutathione reductase can't make reduced glutathione, and if we don't have reduced glutathione, you have oxidative damage from free radicals. So the very high yield disease that I'm talking about is a G6pd deficiency. Now, this is an X-linked recessive disease, and the findings we've already talked about, right? You can't make NADPH, so you get no reduced glutathione. No reduced glutathione means increased oxidative stress. So what else are really high yield findings that you should be familiar with? Well, in a G6pd deficiency, you're going to see what's referred to as Heinz bodies. And Heinz bodies are just little clumps of denatured hemoglobin that are going to accumulate inside of a red blood cell. The more oxidative damage going on inside of a red blood cell means the more cellular components get damaged. So hemoglobin gets damaged and denatures into these little teeny balls that you can see inside of the RBC. That is referred to as a Heinz body. Now, what happens next? Macrophages that are sequestered by the spleen will come out and say, that's weird. What are those little denatured hemoglobin balls doing in the red blood cells? I need to get rid of them. Because after all, a macrophage's job is to eat foreign particles that should not be present in cells. So macrophages from the spleen will bite off little chunks of the red blood cell. And when it does that, all it's trying to do is get rid of that denatured hemoglobin, right? It's trying to get rid of the Heinz bodies. But in doing so, it leaves what are called bite cells. So bite cells, as the name implies, are literally when macrophages from the spleen take bites out of the red blood cells. Now, this is an attempt by the body just to undergo phagocytic clearance, right? All it wants to do is clear out the Heinz bodies, but in doing so, it's breaking the red blood cells and literally taking a bite out of it. So again, big picture, take a step back and pause for a second. Anybody who does not have the enzyme, glucose-6-phosphate dehydrogenase, has by definition a G6pd deficiency. This is an X-linked recessive disease where you lack the key enzyme in the HMP shunt that usually generates NADPH. If you can't generate NADPH and therefore can't make reduced glutathione and therefore can't handle oxidative damage from free radicals in the body, then you're very susceptible to hemolysis when your red blood cells undergo oxidative damage. And it's from those agents that we talked about in the SPIN-F mnemonic that could possibly introduce oxidative stress, because those agents are oxidizing in nature. Now, again, normally a regularly functioning person with this enzyme would be able to handle that stress with no problem. But in those who are deficient in this enzyme, that oxidative insult gets really out of control and you can get really profound anemia. So if on your exam you see a vignette where all of a sudden somebody eats a random bean, right, a fava bean, and they undergo labs or a clinical picture that looks like severe hemolysis, this is your answer. If somebody's started on an antibiotic such as nitrofurantoin or a sulfa drug of any class, and all of a sudden they're having anemia, this is your answer. It's especially high yield to know these pictures because instead of describing what I just said in the clinical vignette, they could very literally just show you Heinz bodies, the denatured hemoglobin, or the bite cells, the attempt at the splenic macrophages to clean up those Heinz bodies, and expect you to know the pathophysiology that we're talking about. So as you can see, glucose-6-phosphate dehydrogenase deficiency is a very high yield biochemical disease. Now, how do you remember all this stuff, right? I've just given you a ton of information, and if anybody has watched any of my videos, you guys know that I don't want to overwhelm you, and my only concern is that you do really well on test day. So I've got a really awesome dirty medicine mnemonic to remember all of this stuff. So when I say G6pd deficiency, or when I think about a G6pd deficiency, I think about 6-GPD. So I just kind of reorder it a little bit. Instead of G6pd, I think of 6-GPD. And what that reminds me of is the number 6 quarter pounder. So if you've ever gone to a Burger King, McDonald's, whatever, you can order, the number 1, the number 2, the number 4, the number 6, whatever. And they have quarter pounders, right? That's a burger in the United States. So I think about quarter pounder. So instead of quarter, I just change that Q to a G. So my mnemonic is take a bite of the number 6 quarter pounder, or quarter pounder, if you will, with Heinz ketchup, right? Heinz is the number 1 ketchup brand in the United States. So take a bite of the number 6 quarter pounder with Heinz ketchup. What does this tell you? Well, bite for bite cells, 6-GPD for G6pd, and Heinz ketchup for Heinz bodies. So this is giving you all the high yield associations and tying it in to the disease. So this is it, guys. This is the slide you need to keep in mind. This is the big picture, right? The combination of the HMP shunt working in tandem in this cyclical way where the wheel keeps spinning, working with the glutathione pathway. Again, The common goal here is to handle free radical and oxidative damage. But individually, the HMP shunt generates NADPH, and the glutathione reductase pathway reduces glutathione. It keeps on working together. There's this beautiful synergy here, and this is the biochemistry that you absolutely need to know. This is very, very high yield, and I think that I've done a good job of explaining it. So watch this video a few times if you need to, but if you're comfortable with all of this material, then you'll be able to answer most, if not all of your questions on USMLE and complex.
خب یکی این فیلم بود، یکی دیگه هم یه فیلم دیگه هستش اگر اجازه بدین این رو هم خدمتتون نشون بدم.
So the glucose molecule is at the center of all metabolism that occurs inside of a human cell. So when the glucose molecule enters inside of cells, these glucose molecules go through a pathway referred to as glycolysis. And in glycolysis, we take glucose molecules and we do a bunch of chemical reactions, eventually taking those glucose molecules and converting them into pyruvate molecules. And keep in mind, each of these chemical reactions are catalyzed by their own unique enzymes. So now, once we go through glycolysis and form these pyruvate molecules, then they can enter inside of the mitochondria. And in the mitochondria, we take these pyruvate molecules and convert them into acetyl-CoA molecules. Now, once we form these acetyl-CoA molecules, they can react with oxaloacetate molecules to form citrate molecules. And once we form these citrate molecules, again, we can go through a bunch of chemical reactions, eventually reforming oxaloacetate molecules, which can react with new acetyl-CoA molecules to form new citrate molecules to go through this cycle over again. So this pathway is referred to as the Krebs cycle. But these two pathways, glycolysis and the Krebs cycle, are collectively referred to as central metabolism because glycolysis and the Krebs cycle are at the center of all metabolism that occurs in the cell. And nearly all cells in the human body go through central metabolism. Why? Well, when we take glucose molecules to go through central metabolism, we create ATP molecules. And we know these ATP molecules are high energy molecules, which are used as a source of energy as a source of fuel to energize all the energetic chemical reactions the cell needs to survive. But not just that, when we take glucose molecules to go through central metabolism, we also create these reduced cofactors like NADH and FADH2. And we know these reduced cofactors have electrons that can be donated to the electron transfer chain to create even more ATP. So the point is, when we go through central metabolism, we create the ATP that's needed for the cell to survive. But what's interesting is, yeah, we can take glucose molecules to go through central metabolism, but we can also take other monosaccharides to enter central metabolism. For example, galactose, we can take galactose and convert it into this particular intermediate. Now, once we form this particular intermediate, it swoops in to central metabolism and can be used to create ATP. So the point is, any molecule that can be converted into one of these intermediates can simply swoop in and be used to create ATP. And lots of different monosaccharides can be converted into intermediates and be used to create ATP. So what about proteins and their component amino acids? Can we take these amino acids to enter central metabolism to create ATP? Well, it depends, because keep in mind, amino acids have a carboxyl group and an amine group. But notice, none of these intermediates of central metabolism have nitrogen molecules. So therefore, if we want to take amino acids to enter central metabolism, we must get rid of this nitrogen group. But we can do that. We can get rid of this nitrogen group and replace it with the carbonyl group. Once we do that, now we have this carbon backbone, and these carbon backbones can enter central metabolism. And notice alanine, when we get rid of the amine group and replace it with this carbonyl group, we form this molecule, which happens to be pyruvate. So this can simply slip in as pyruvate and enter central metabolism and be used to create ATP. So this is true for any generic amino acid with R group. And again, it's the same idea. We get rid of the amine group, replace it with the carbonyl group, and now we have this carbon backbone, which we refer to as an alpha ketoacid. And we know different amino acids will have different R groups, but depending on what that R group is, we can do chemical modifications to convert these ketoacids into intermediates of the central metabolism, and therefore enter central metabolism and be used to create ATP. But you might wonder, when we take these amino acids and get rid of this amine group to form these carbon backbones to enter central metabolism, what do we do with these amine groups? Because we know ammonia is toxic to the cell. So what do we do with all this ammonia? Well, we usually have a nitrogen acceptor, and the universal nitrogen acceptor is alpha ketoglutarate. So this alpha ketoglutarate accepts that nitrogen. So this nitrogen waste we donate to alpha ketoglutarate to form glutamate. Now, depending on what the cell was, there will be this nitrogen transporter where we can take this nitrogen from glutamate, donate it to the nitrogen transporter. Then this nitrogen transporter can enter the blood and transport that nitrogen to the liver hepatocytes, where then that nitrogen is donated to another alpha ketoglutarate to form glutamate, which can then safely enter the urea cycle, where we take that nitrogen and convert it into the safe urea, which can safely be disposed out of our body. But only the liver can safely dispose of nitrogen. So the point is, if we have a tissue that wants to use proteins and amino acids as a source of energy, we must get rid of that nitrogen group. But how do we get rid of that nitrogen group? Well, again, we donate that nitrogen, which goes to the nitrogen transporter, which can then safely donate that nitrogen to the liver, and the liver can safely dispose of that nitrogen through the urea cycle. So what about fatty acids? Can lipids and fatty acids enter central metabolism and be used to create ATP? Well, it's a little complicating. So if we want to use fatty acids to create ATP, we take the fatty acids and go through a path that we refer to as beta oxidation. And every time we go through a round of beta oxidation, we create an FADH2 and an NADH, and we lose 2 carbons in the form of acetyl-CoA. So that happens every time we do a round of beta oxidation. So now we have a fatty acid that's 2 carbons shorter. Now once we form this fatty acid, we can do another round of beta oxidation. And again, every time we do a round of beta oxidation, we create these reduced cofactors and we lose 2 carbons in the form of acetyl-CoA. So we know these reduced cofactors can directly fuel the electron transport chain to create ATP. So therefore, these fatty acids, the way they are used to make ATP is directly by forming these reduced cofactors to fuel the electron transport chain. And again, we also create these acetyl-CoA molecules, which theoretically could enter the Krebs cycle, but in reality, it's a little more complicated. So we'll talk about that later. So we've already learned when we take glucose molecules and go in this direction to form pyruvate molecules, that pathway is referred to as glycolysis. However, we can also go in the reverse direction. We can take pyruvate and go in the reverse direction, forming glucose molecules. And we can do this because each of these chemical reactions were reversible. However, once we take pyruvate and form acetyl-CoA, this is irreversible. This is a one-way street, and we cannot go in the reverse direction. Once we form acetyl-CoA, we cannot go in the reverse direction to form pyruvate. However, pyruvate, we can go in the reverse direction to form glucose molecules. So when we go in this reverse direction, it's referred to as gluconeogenesis, the genesis of new glucose molecules, where we go in this reverse direction and we create new glucose molecules. So in certain contexts, we need to go in this reverse direction to form new glucose molecules. However, glucose is made out of 6 carbons. So if we want to make glucose molecules, we need a source of carbons. So what are those major sources of carbons? Well, lactate has 3 carbons. So lactate can be converted into pyruvate. And when we form pyruvate, we know we can go in the reverse direction to form glucose molecules. So therefore, these 3 carbons in lactate can be used to form glucose molecules. The same thing with glycerol. We know we have these triglycerides, which is a storage form of energy. But when we hydrolyze these ester linkages, and release a glycerol molecule. This glycerol molecule has 3 carbons, and glycerol can be converted into this intermediate, which we know can then be used to make glucose molecules. However, what about ketoacids, like beta-hydroxybutyrate? Can these atoms be used to biosynthesize glucose molecules? Well, alpha ketoacids can be converted into acetyl-CoA. But we know once we form acetyl-CoA, we cannot go in this reverse direction to form pyruvate molecules, so we're stuck. Once we form acetyl-CoA, we are forced to go in this direction. So therefore, these atoms in ketoacids cannot be used to form glucose molecules. So what about proteins and these amino acids? Can we take these atoms and amino acids to form glucose molecules? Well, it depends. For example, alanine, we know when we get rid of the amine group, we form this alpha keto acid, which happens to be pyruvate. So we know this can enter as pyruvate, which can be used to make glucose. So therefore, these atoms in alanine can go through this pathway to form glucose molecules. What about aspartate? Well, we know we need to get rid of the amine group, so now we form this alpha keto acid. But this particular alpha keto acid, just by chance, happens to be oxaloacetate. And something you just have to memorize is just by chance, we happen to have an enzyme that can take oxaloacetate to form pyruvate. And once we form pyruvate, we know we can form glucose molecules. So therefore, aspartate can go through this pathway to form this intermediate, which is again this intermediate, which can then be used to form glucose molecules. But what about leucine, the amino acid leucine? Well, again, we get rid of the amine group, so now we form this alpha keto acid. But this alpha keto acid, we have the enzymes to convert it into acetyl-CoA. And we know acetyl-CoA cannot be used to form glucose molecules. We know it cannot go in this reverse direction. It's forced to go in this direction, so it can't be used to form glucose molecules. But you might wonder, can't we take acetyl-CoA to go in this direction to form this guy to then be used to form glucose? Well, no. Acetyl-CoA has theoretically 2 carbons that it could donate to form glucose molecules. However, once acetyl-CoA goes through this pathway, it loses 1 carbon as carbon dioxide, then it loses its 2nd carbon. So therefore, these 2 carbons are lost as carbon dioxide by the time it forms this intermediate. So therefore, these carbons cannot be used to form glucose molecules. But what about methionine? Well, again, we get rid of the amine group, so we form this alpha keto acid. And this particular alpha keto acid can be converted into this particular intermediate, which again can enter this pathway and be used to form this molecule. So therefore, amino acids can be used to form glucose molecules. And the amino acids that can be used to form glucose molecules are referred to as the glucogenic amino acids. However, other amino acids cannot be used to form glucose molecules, so they're referred to as ketogenic because they can be used to form ketone bodies. So what about fatty acids? Can these atoms and fatty acids be used to create glucose molecules? We know when we go through rounds of beta oxidation, we create acetyl-CoA molecules. And we already explained these carbons in acetyl-CoA can be used to form glucose molecules because those carbons are lost as carbon dioxide. However, if you happen to end up with 3 carbons in your final fatty acid, this just by chance can be converted into this intermediate, and it can be used to form glucose molecules. So these last 3 carbons can be used to form glucose molecules. So these 2 carbons in acetyl-CoA are incredibly important. And yeah, even though these 2 carbons cannot be used to form glucose molecules, they can be used for other purposes. For example, we can take these 2 carbons in acetyl-CoA to biosynthesize free fatty acids like palmitic acid through the process of fatty acid synthesis. Now, once we form this palmitic acid, it can react with a moiety of glycerol, where we take 1 moiety of glycerol, 2 moieties of palmitic acid, and 1 inorganic phosphate to form these phospholipids. And we know these phospholipids are key components of our cell membranes. But not just that, we can take 1 moiety of glycerol and 3 moieties of palmitic acid to form triacylglycerides through the process of lipogenesis. And we know these triacylglycerides are the primary storage form of energy our bodies use. For example, when we're in the fed state, so we've just eaten a big meal, we know when we eat a big meal, our bodies will have lots of glucose, proteins, and fatty acids that we got from our meal. So we want to store those molecules. For example, all those glucose molecules, we enter through a process of glycogenesis, where we take those glucose molecules and store them in the form of glycogen, which we know is just a storage form of glucose molecules. So we take excess glucose molecules and store them in the form of glycogen. And all those excess proteins and those amino acids we use to replace already existing enzymes and structural proteins and peptide hormones. However, eventually we're going to fill up our glycogen stores, where we filled up our glycogen stores and we've replaced all of the enzymes and structural hormones and glucose and proteins into triacylglycerides as a storage form of energy. So we know these triacylglycerides is a primary storage form of energy our bodies use. The atoms in serine and reacted with these atoms in palmitic acid to this sphingosine backbone. And we know the sphingosine backbone is very important to form other important lipids. For example, we take the sphingosine backbone, we react a free fatty acid to this amine group, or we add a free fatty acid to this amine group to form ceramide, the ceramide molecule. And we know with the ceramide molecule, we can use it to form lots of important molecules. For example, we take ceramide and we add a phospho group to this hydroxyl head group to form sphingomyelin. And sphingomyelin is a key component of the myelin sheaths of our neurons. Also, what we do is we can take the ceramide and we can add a carbohydrate to this hydroxyl head group to form cerebrosides. Also, we can add more carbohydrates to form gangliosides. So that's what happens during the fed state. But what about the fasted state? For example, let's say we haven't eaten a meal for a couple days. What is the source of energy if we don't eat? Well, we can use these triacylglycerides as a storage form, as a source of energy, where we hydrolyze these ester linkages, releasing free fatty acids. And we know we can take free fatty acids and go through rounds of beta oxidation. And every time we do a round of beta oxidation, we create reduced cofactors and acetyl-CoA molecules. And we know these reduced cofactors can fuel the electron transfer chain to create ATP. So this is one way we create ATP during a fasted state. We use our triacylglycerides, our storage form of energy, as a source of energy. But we know when we go through rounds of beta oxidation, we also create these acetyl-CoA molecules. So what do we do with these acetyl-CoA molecules? Well, we can react three CoA molecules with each other to form this HMG-CoA. And this HMG-CoA is a very important molecule because in the liver, in hepatocytes, we can take HMG-CoA and go through a process referred to as ketogenesis, where we form these keto bodies like beta-hydroxybutyrate and acetoacetate. So during a fasted state, with the fasted state hormones and with a lot of acetyl-CoA molecules that we get from beta oxidation, we can form HMG-CoA and go through ketogenesis to form these ketoacids in the liver. But why is the liver forming all these ketoacids? Well, these ketoacids can leave the liver, enter the blood, and enter into other tissues. And these other tissues can take these ketoacids and convert them into acetyl-CoA molecules. And then these tissues can use these acetyl-CoA molecules to enter the Krebs cycle to create reduced cofactors to fuel the electron transfer chain to create ATP. So this is what happens during the fasted state. However, this HMG-CoA is really important and can enter other pathways. So again, in a fasted state with the hormones during a fasted state, we take HMG-CoA to form ketoacids through ketogenesis. But in other contexts, for example, during a fed state, we can take HMG-CoA and use it to form these 5 carbon units. These 5 carbon units, which can be used as building blocks to build larger organic carbon-based molecules. For example, take 2 of these 5 carbon units, react them with each other to form a 10 carbon unit. Then we take a 10 carbon unit, react it with the 5 unit to form a 15 carbon unit. Then we react two of those 15 carbon units to form a 30 carbon unit, which is referred to as squalene. So this squalene has free rotation among some of these bonds, so the squalene can fold and get in a particular conformation react with itself to form cholesterol, which can then be to form cholesterol. Notice this is pretty interesting. As long as we have glucose molecules, we can take these atoms and glucose molecules to form acetyl-CoA molecules, which can form HMG-CoA, which can form these 5-carbon units, which can be used to form larger organic molecules like cholesterol. So we can biosynthesize cholesterol. And that's good because we know cholesterol is super important and is used to create lots of important hormones. For example, we can take cholesterol, react it with some energy from sunlight, and then react it with the lithium different derivatives of vitamin D, which is important. We can cholesterol and enter and use mix of other important steroid hormones. For example, in the testes, we can go through this pathway to form testosterone. In the ovaries, we can go through this pathway to form estradiol. Different parts of the adrenal cortex go through different pathways to form cortisol and aldosterone. These important hormones. And in the liver, we can take cholesterol to form these bile salts, which are needed to emulsify and absorb fats from our diet. But what's pretty cool is we can take these intermediates of central metabolism and use them to form amino acids. But we know amino acids have nitrogens, and none of these intermediates of central metabolism have nitrogens. So therefore, to take these intermediates to form amino acids, we need a source of nitrogen. And that nitrogen usually comes from glutamate, which is the universal nitrogen donor. So what are some examples? Well, we can take oxaloacetate, which is an intermediate of central metabolism. So we can take this oxaloacetate and use it to form aspartate. And we can do that because glutamate has a nitrogen that it donates to oxaloacetate to form aspartate. So that's pretty cool. We took this intermediate of central metabolism and formed aspartate. Another example is we can take this particular intermediate of central metabolism and form alanine. And again, we know we were able to do that because we have glutamate, which can donate nitrogens to these intermediates of central metabolism to form certain amino acids. So that's pretty cool. We can take these intermediates of central metabolism to biosynthesize certain amino acids. And here's a comprehensive list of all the amino acids we can form. For example, this intermediate of central metabolism can be used to form serine, which can be used to form cysteine and glycine. This intermediate can be used to form aspartate, which can be used to form asparagine. And also, all these amino acids with these red stars, we make with very low efficiency. We take this intermediate and form phenylalanine with very low efficiency. So therefore, most of these essential amino acids we need to get from our diet. But a lot of amino acids we can biosynthesize, which is pretty neat. And we know these amino acids are super important to form proteins, to form enzymes and hormones and transcription factors and etc. So we already explained how we can take this intermediate of glycolysis to form serine. But this serine amino acid is really important because it has this carbon, which is used as a carbon donor to build larger molecules. So how do we do this? Well, we take this carbon and we donate it to tetrahydrofolate to form this methylated form of tetrahydrofolate. Then we can take this carbon to donate it to homocysteine. So now homocysteine gains that carbon, methionine. Now methionine reacts with ATP to form this S-adenosylmethionine, this SAM molecule, which is the universal methyl donor in the body. So again, if we want to build molecules, we need carbons. So a lot of the times those carbons come from SAM. And also if we want to methylate DNA, those carbons come from SAM. So this SAM is the universal methyl donor where it has these carbons that donate for anabolism. But also mind, this tetrahydrofolate also has a carbon that can donate for anabolism. So these are the two universal carbon donors, two universal methyl donors. What's also, we can take certain amino acids and other sulfur-containing compounds from our diet and use them to form sulfate. Once we form sulfate, we can react it with ATP using this enzyme to form this particular intermediate. Then we can react it with another ATP molecule using this enzyme to form this 3-phosphoadenosine 5-phosphosulfate, this PAPS molecule, which is a universal sulfur donor. So if we need to build molecules that need sulfur molecules, they usually come from this universal sulfur donor. So for example, we know there are lots of different monosaccharides our bodies use for lots of different purposes. For example, we sometimes glycosylate lipids. So we need different monosaccharides to glycosylate these lipids. Also, proteins get glycosylated. So these proteins get glycosylated with lots of different monosaccharides. And we also know red blood cells have proteins that get glycosylated, and that's the basis of blood type. So we know there are lots of different monosaccharides that our bodies use for lots of different purposes. But how do we create these monosaccharides? Well, usually they come from these intermediates of glycolysis. And if you're interested, here are the pathways. I know this looks really complicated, but the point is we can take this intermediate of glycolysis to go through this pathway to form all of these monosaccharides. And again, notice this monosaccharide has a sulfur. Where does it get that sulfur? Well, again, we already explained PAPS is a universal sulfur donor. So once we have this intermediate, we take this PAPS to donate a sulfur to form this sulfated glucosamine. And also we can take this intermediate of central metabolism and throw on a nitrogen from glutamine to form this monosaccharide with a nitrogen group, which can then enter different pathways to form lots of different monosaccharides. And again, whenever a monosaccharide has a sulfur, you know, got it from PAPS. So I know this looks complicated, but the point is there are lots of different monosaccharides we can biosynthesize, and usually they come from these intermediates of central metabolism. We can also take this particular intermediate of glycolysis to enter a pathway referred to as the pentose phosphate pathway. And in this pentose phosphate pathway, we produce two important products. One, we create ribose 5-phosphate, and also we create these reduced NADPH cofactors. And these reduced NADPH cofactors are really important because they have electrons. And essentially, they can use these electrons and these hydrides as a source of electrons for anabolism. Because we know if we want to build larger molecules, we need electrons to form these bonds. So these electrons usually come from NADPH, which has electrons. But also, these NADPH can take their electrons and donate them to this oxidized glutathione to form reduced glutathione. And this reduced glutathione is an endogenous antioxidant. This is an antioxidant. So if we have reactive oxygen species and if we have free radicals, we know these are toxic molecules. They're super reactive because they have these unpaired electrons. So they're toxic. They react with biomolecules and destroy them. So these reactive oxygen species and free radicals are toxic. However, fortunately, we have this endogenous antioxidant, this glutathione, to donate electrons. So now this unpaired electrons gets paired to form a neutralized safe molecule. So that's pretty neat. Essentially what we can do is we can take these electrons from glucose, go through this pathway, take those electrons, and eventually donate them to NADPH, which can donate those electrons to glutathione to form an antioxidant, which can then take those electrons to donate them to free radicals to neutralize those free radicals. So we have this endogenous antioxidant system, which is pretty neat. And we know this is important because our electron transport chain in our mitochondria is constantly producing free radicals. Even if you're healthy, young, whatever, our mitochondria are constantly producing free radicals, which get metabolized to other free radicals. And these free radicals, for example, hydroxyl radical, is extremely dangerous and toxic. It can react with free fatty acids, which react with these hydroxyl radicals to form this peroxy radical, which goes through a very nasty free radical chain reaction, which forms more free radicals and can form carcinogens, and this is a very toxic pathway. However, fortunately, we have this reduced glutathione, which got its electrons from glucose. So this reduced glutathione is an antioxidant. It can donate electrons to vitamin C, which can then donate that electron to vitamin E, which can then donate that electron to this peroxy radical, stopping that toxic chain reaction. So that's pretty neat. We have this endogenous antioxidant system. So it's important to have a strong endogenous antioxidant system by having lots of vitamin C and vitamin E, and also having the right minerals that's needed for these enzymes in this pathway. For example, selenium and magnesium and copper and zinc, these are important minerals to allow a very strong endogenous antioxidant system. However, what about this ribose 5-phosphate? Well, we can take this ribose 5-phosphate and we can add a nitrogenous group to form this nucleic acid precursor, where these atoms came from aspartate, and these atoms came from glutamate to form this nucleic acid precursor, which can be used to form pyrimidines like cytidine and uridine, these nucleic acids. So that's pretty cool. And another example is we can take this ribose 5-phosphate, add a different nitrogenous group to form this nucleic acid precursor, where these atoms came from glycine, these atoms came from glutamate, and these atoms came from aspartate. And then these two carbons came from tetrahydrofolate, which is a methyl donor. But then we form this intermediate, which we know can be used to form purine nucleic acids like adenosine and guanosine. So that's pretty cool. We can biosynthesize these nucleic acids as long as we have glucose to go through this pathway to form ribose 5-phosphate, which makes this component, and as long as we can form these amino acids to donate the atoms for this nitrogenous base, we have everything we need from central metabolism to biosynthesize nucleic acids. So that's pretty cool. But also we can degrade these nucleic acids. For example, GTP, we can degrade it where we get rid of the phosphate groups, then we hydrolyze this bond, releasing this nitrogenous base, which we can then catabolize to form uric acid, which can safely be disposed in our urine. And the same thing with the pyrimidines. We can degrade these pyrimidines to form these end products, which can, for example, CO2 we can simply breathe out. Ammonia gets converted into urea, which can be safely exposed of, and these compounds can also be safely exposed of. So we can biosynthesize these nucleic acids, but we can also safely degrade them and get rid of these waste products. We can also use these intermediates of central metabolism to biosynthesize neurotransmitters. For example, we can take this intermediate to form a phenylalanine. And the truth is, we do this with a very low efficiency, so most of the phenylalanine we need from our diet. But once we have phenylalanine, we can form tyrosine, which can be used to form L-dopa, which can be used to form dopamine. So that's how we biosynthesize dopamine. But we can also go a step further if we need norepinephrine. And we can also go another step further if we need epinephrine. So this is how we biosynthesize a lot of neurotransmitters. There are other pathways. For example, we take acetyl-CoA and react it with this choline molecule to form acetylcholine, which is a really important neurotransmitter. We take this particular intermediate to form glutamate, which can be used to form GABA and glutamine. These are two important neurotransmitters. We can form tryptophan with very low efficiency, so most of the tryptophan we need from our diet, but once we have tryptophan, we can then form serotonin, or go a step further to form melatonin. And we can also take histidine to form histamine. So that's how we biosynthesize a lot of these important neurotransmitters, but a lot of them come from these intermediates of central metabolism. Also, we can take this intermediate to eventually form glycine, which we can react with succinyl-CoA to form this particular intermediate. Then we take two of these guys and react them with each other to form this compound. Then we take four of these guys, react them with each other to form this porphyrin ring. And we know this porphyrin ring with this iron group can be used, this porphyrin ring can be used to form hemoglobin or myoglobin, or the cytochromes important for metabolism, or the electron transfer chain. So that's pretty cool. We can form this porphyrin ring from these intermediates of central metabolism. So now you can see why glycolysis and the Krebs cycle are collectively referred to as central metabolism, because these two pathways are at the center of all metabolism that occurs in the cell. All these different metabolic pathways stem from these intermediates of central metabolism. And if you're interested, in the link below, I have a different link for each of these pathways in more detail. For example, I have a separate link dedicated for amino acid biosynthesis, and another link dedicated to ketogenesis, and another link dedicated to fatty acid synthesis. I have different links dedicated to each of these chemical reactions. So one last final note. Yeah, we can take these intermediates of central metabolism to biosynthesize lots of important compounds. However, there are limitations to central metabolism. For example, by definition, we cannot biosynthesize vitamins. We cannot take glucose and these intermediates to biosynthesize vitamins. For example, vitamin B3 niacin, yeah, this may be a very simple molecule. However, we simply don't have the enzymes to take these intermediates to biosynthesize niacin. So therefore, this vitamin we need to get from our diet. So that's true for all vitamins. vitamins we must get from our diet. And the same thing with minerals, with electrolytes. For example, sodium and potassium and chromium. We cannot take these intermediates. We can't take glucose with these carbons and oxygens and hydrogens and transmutate these elements to form sodium ions or potassium ions. So therefore, these minerals we must get from our diet. So all these elements in purple are the minerals we must get from our diet. And these minerals are really important. Some of these minerals, for example, chromium is really important for certain enzymes to function properly. So there are limitations to what we can do with central metabolism.
خب مجدد سلام عرض میکنم خدمت همه شما. این فیلمایی رو که ملاحظه کردید مربوط میشه به درک بهتر شما از درس، چون من دیدم دوباره این مطالب رو اومده داره توضیح میده، مخصوصا ارتباط مسیرهای مختلف متابولیسمی رو. برای همین این دو تا فیلم رو، حالا اولیش که مربوط میشد به مسیر پنتوز فسفات و عملکرد گلوتاتیون ردوکتاز که یکی از آنزیم های بسیار مهمی هست که به انسولین به مقدار بسیار زیاد، عذرخواهی میکنم، به NADPH نیاز داره، راجبش صحبت میکرد، اون رو براتون گذاشتم. و این یکی هم که دومی فیلم هم که حالا یه مقداری حجمش هم زیاد بود، مربوط میشه به مسیرهای متابولیسمی که همه اینها به همدیگه مرتبط هستن. در مورد مطالبی که جلسه پیش خدمتتون عرض کردم، من اینجا توی لیست نوشتم که تاثیر انسولین توضیح دادم خدمت شما. من این صفحاتش رو برای شما هم گذاشتم، یه مقداری تصویر رو بزرگترش میکنم، چون مال خود، البته خود اسلایدم هست، ولی من دیگه مال خود کتاب رو بزرگش میکنم. ببینید، ما جلسه گذشته در مورد مسیرهای مختلف متابولیکی و ارتباطشون با هم دیگه و تعداد زیادی از مولکول‌ها که درست تو نقطه انشعاب قرار میگیرن خدمتتون صحبت کردم. بعد رسیدیم به پیام‌های هورمونی که اینها در واقع میتونن متابولیسم کربوهیدرات‌ها، لیپیدها و پروتئین‌ها رو ادغام کنن. متوجه نمیشم تصویر، نمیدونم بقیه هم تصویر رو واضح نمیبینیم، تصویر واضح نیست. من این عکسیه که از خود کتاب گرفتم. من متاسفانه که از دانشجوها هم فایلش رو که از من میخواست، من فایلی کتاب رو ندارم، عکس رو میفرستم. واضح هست آقای شویلی؟ متشکرم، خیلی ممنون، مرسی، متشکرم، ممنون. در مورد انسولین، خب انسولین رو ما میتونیم که یک هورمون آنابولیک هست. متشکرم، خیلی ممنون، مرسی، که ذخیره گلوکوز رو میتونه در بدن تحریک بکنه. بعد خود انسولین گلیکوژنز و ساخت اسیدهای چرب در کبد، ساخت تریگلیسیرید تو بافت چربی، جذب گلوکوز و گلیکوژنز رو توی بافت ماهیچه میتونه اینها رو تحریک بکنه. تک تک عملکرد هورمون انسولین در بافت‌های مختلف رو شما اینجا میتونید ببینید، توی بافت کبد، تو بافت ماهیچه. بعد تأثیری که میتونه داشته باشه روی بافت چربی و ارتباطش با بافت کبد رو میتونید ملاحظه بفرمایید. یک شکل جامع و کاملی هست که نقش هورمون‌ها در ذخایر سوختی، در حرکتشون، در توزیعشون رو اینجا برای شما مشخص کرده. هورمون بعدی هورمون گلوکاگون هستش. اگه اجازه بدید من بیارم صفحه بعدی که توضیحات شما ببینید. اونقدر توی کتاب هم راجع به نقش گلوکاگون خیلی سریع گذشته. من انتظار داشتم بیشتر توضیح بده، ولی دیدم که نه، خیلی کوتاه گفته. نمیدونم بقیه دوستانتون که آقای شویلی میگن تصویر واضحه، به نظر منم خوبه، یعنی شما میتونید، مشکلی نداره. کتاب رو که من متاسفانه فایلش رو ندارم براتون بفرستم. بله، متشکرم، مرسی. گلوکاگون رو به عنوان یکی از هورمون‌های کاتابولیک حساب می‌کنن. گلوکاگون میتونه باعث افزایش میزان گلوکوز به خون بشه. بعد مخصوصاً بین وعده‌های غذایی که یه جورایی تنظیم کننده گلوکوز خون هم هست. گلوکاگون هم فرایند گلیکوژنولیز رو تحریک میکنه، هم فرایند گلوکونئوژنز رو تحریک میکنه. بعد در عین حال تجزیه پروتئین های ماهیچه و استفاده از اسکلت های کربنی اسیدهای آمینه رو میتونه به عنوان سوبستراها در مسیر گلوکونئوژنز توی کبد این رو فراهم بکنه، میتونه مهیا بکنه. در حالی که مثلا از این طرف دیگه اگر بخوایم روی لیپولیز توی بافت چربی بخوایم متمرکز بشیم، مثلا این هورمون گلوکاگون کار دیگه که انجام میده این هستش که میتونه مثلا لیپولیز تو بافت چربی رو افزایش بده. بعد به تبع اون هم گلیسیرول فراهم میشه برای تبدیل به گلوکوز یا اسید چرب و در این حال انرژی لازم رو هم برای بدن میتونه مهیا بکنه. قسمت بعدی کاتکولامین ها هستن. کاتکولامین ها خب مثل گلوکاگون با تحریک آزادسازی گلوکوز به خون درست مسیر عکس انسولین رو پیش میرن. بعد میتونن به صورت مستقیم هم گلوکونئوژنز رو تو کبد، هم گلیکوژنولیز رو تو کبد، هم تو بافت چربی فرایند لیپولیز رو، بعد آزاد کردن اسیدهای آمینه و لاکتاز رو توی ماهیچه‌ها میتونن تحریک بکنن. پس میبینید که شاید ما بیشتر تو مسیرهای متابولیسم انسولین و گلوکاگون رو انقدر بهش توجه میکنیم، کاتکولامین ها در جایگاه خودشون بسیار مهم هستن. علاوه بر این کاتکولامین ها میتونن باعث آزاد شدن گلوکاگون به وسیله سلول‌های آلفا توی لوزالمعده بشن و کار دیگه که انجام میدن باعث مهار آزادسازی انسولین از سلول‌های بتا میشن. پس بنابراین یکی از مهمترین کارهایی که انجام میدن اینه که سطح هر دو تا هورمون رو میتونن تنظیم بکنن یه جورایی. توی این فصل از کتاب این قسمت انشعابات مسیرهای متابولیسمی تقریباً به اتمام رسید. وارد فصل بعدی میشیم که فصل بعدیش انتقال لیپیدها هست که به صورت ساختار لیپوپروتئین ها انجام میشه به همراه عملکرد و متابولیسمشون. احتمالا شما مطالبی که مربوط به لیپوپروتئین ها هست یک مقداریش رو در قسمت بیوشیمی ساختار باید خونده باشیم. بله درسته. صفحات شاید بعضی از تصاویری که گذاشتم به خاطر حالا چون عکس دارم میگیرم بعد خواستم وضوحش هم حفظ بشه، نورش هم بنز کافی باشه، یه مقداری کجه. مثلا خود همین این رو بله یه مقداری کج هستش دیگه. نمیتونستم بهتر از این گوشی موبایل رو روی صفحه کتاب زومش کنم، چون از این طرفی می اومدم تصویر رو زوم بکنم بعد مجبور بودم توی زاویه هم بگیرم که نور خیلی خوب باشه. خود صفحات کتاب هم یک مقداری تیره است. بعد حالا باز این تصاویری که شما میبینید مقداری نورش رو بیشتر کردم شاید بهتر بشه. خب میریم قسمت بعدی، لیپوپروتئین های پلاسما. ببینید لیپوپروتئین های پلاسما از جمله کمپلکس های بزرگ هستن که توی آب محلولن. بعد انتقال لیپیدهای نامحلول رو بر عهده‌شونه که در خون بتونن اینها رو جدا بکنن. لیپوپروتئین ها از یک قسمت مرکزی به وجود اومدن که حالا شامل تریگلیسیریدها ها هست، شامل استر کلسترول هست. یه قسمت پوشاننده هم داره که به صورت تکلایه است. هم توش پروتئین داره که شما میتونید این آپوپروتئین ها رو ببینید، هم توش ساختار فسفولیپیدی داره بعد کلسترول های فری داره، کلسترول های داره که به صورت استر در نیومدن. بعد خود همین ها یک ساختار خاصی رو میتونه برای لیپوپروتئین ها ایجاد بکنه. اجزای پروتئینی هم معمولا به صورت آپوپروتئین هستند یا آپولیپوپروتئین، اینها رو به این صورت نامگذاری میکنن. ساختار کلی لیپوپروتئینی رو میتونید اینجا داشته باشید. بعد لیپوپروتئین ها رو بر اساس حالا چگالیشون به 5 دسته بزرگ تقسیم بندیشون میکنن. یکی ما کیلومیکرون ها رو داریم، بعد لیپوپروتئین هایی داریم به نام very low density لیپوپروتئین هایی با چگالی بسیار پایین VLDL. بعد لیپوپروتئین هایی داریم با چگالی متوسط که اینجا نذاشته به نام IDL هستن. لیپوپروتئین هایی داریم به نام low density لیپوپروتئین با چگالی پایین و لیپوپروتئین هایی که چگالی بالا دارن های دنسیتی لیپوپروتئین باز از دسته دیگری از لیپوپروتئین ها هستن. یک موردی که وجود داره دانسیته یا چگالی تقریبا با غلظت تریگلیسیرید نسبت عکس رو داره، در حالی که با غلظت پروتئین نسبت مستقیم رو داره. یعنی هر چقدر میزان غلظت پروتئین افزایش پیدا کنه میزان دانسیته هم یا چگالی هم افزایش پیدا میکنه، با هم یک نسبت مستقیم دارن. در شیلومیکرون ها حدود 1 تا 2% ساختار پروتئینی هست، در حالی که مثلا در مورد تریگلیسیریدشون رو اگر نگاه کنیم 98 تا 99% ساختار شامل تریگلیسیرید هست. اینها از لحاظ میزان درصد پروتئین ها و تریگلیسیرید ها با همدیگه بسیار متفاوت هستن، چگالیشون پایینه. ولی مثلا وقتی که میان روی HDL مطالعاتشون رو انجام میدن میبینن که 50%شون پروتئین هست، 50%ش لیپید. خب این نصفش در ساختارش پروتئین وجود داشته باشه، این بسیار جزء چربی های مطلوب محسوب میشه که در داخل خون وجود داره. توی کتاب مطرح میکنه مربوط میشه به یک جدولی که خصوصیات لیپوپروتئین های پلاسما رو اینجا براتون همه رو نوشته. اینا برای حفظ کردن نیست. درصد هر کدوم از اجزای لیپوپروتئینی، پروتئین ها، لیپید ها، تریگلیسیرید، کلسترول استریفیه شده، بعد خود کلسترول، بعد فسفولیپید، همه درصدش رو مشخص کرده که اینا فقط برای اطلاعات عمومی شما هست. یه نکته دیگه که باید در نظر بگیرید این هستش که هر چقدر به سمت لیپوپروتئین با چگالی بیشتر حرکت میکنید توی جدول، قطر هر لیپوپروتئین هم کاهش پیدا میکنه. برای جدا کردن لیپوپروتئین ها از سانتریفیوژ استفاده میشه که بر اساس چگالی هم این جدا سازی صورت میگیره. موقع سانتریفیوژ سرعت شناور شدن هر لیپوپروتئین در محلول نمکی به میزان تقریبا تریگلیسیرید بستگی پیدا میکنه. مثلا ما اگر بخوایم به همه اینها رو با هم مقایسه بکنیم، کیلومیکرون ها سریعتر از همه شناور میشن، در حالی که مثلا HDL شناوری کمتری داره. شما توی اینجا هم میتونید این رو ببینید. بعد پس بنابراین یک ارتباطی بین حرکت الکتروفورتیک و سرعت شناوری وجود داره توی آزمایشگاه‌های بالینی که ازش استفاده میکنن. حالا توی اینجا هم اومده البته از الکتروفورز استفاده کرده، هم از سانتریفیوژ استفاده کرده. بعد مثلا توی این تصویری که شما سمت چپ می‌بینید مهاجرت الکتریکی رو توی pH 8.6 نشون داده. مثلا کیلومیکرون ها تو قسمت بالا قرار میگیرن، بعد مثلا HDL تو پایینه. بعد این بتا و آلفا 2 و آلفا 1 هم مربوط میشه به گلوبین ها که حالا اومده با اینها یک مقایسه هم اومده انجام داده. بعد پس بنابراین کلسترول سرم و میزان تریگلیسیرید پلاسما برای یادم نمیاد عکسشو گرفتم، نه نذاشتم. ما 4 نوع آپوپروتئین داریم توی ساختار لیپوپروتئین ها. هم آپو A داریم، B داریم، C داریم، E داریم. انواع آپوپروتئین ها همشون هم توی کبد ساخته میشه. مثلا توی سلول های روده ای ما فقط آپو A و آپو B رو داریم. بعد خیلی روی این زیرواحد ها کار کردن، ولی باز عملکرد دقیق آپوپروتئین ها اونقدر مشخص نیست. اومدن روشون کار کردن، دیدن مثلا یه مقداری وظایف دینامیکی هم دارن. حالا وقتی که تصویر خود کتاب رو فکر میکنم اگر اجازه بدین من یه چک بکنم، نذاشتمشون. انشاءالله برای جلسه آینده صفحات کتاب رو میذارم، بعد داخل اونها هم میتونید ببینید. مثلا آپوپروتئین ها رو جزء ساختار ضروری لیپوپروتئین ها اینها رو محسوب میکنن. ما آپو A رو داریم، B رو داریم، C رو داریم، E رو داریم و بعد اینها جزء عملکردهای بسیار مهمشون به طور مثال انتقال کلسترول هست. بعد مثلا آپو B کارش اتصال به LDL رسپتوره، یا مثلا آپو C کارش تنظیم لیپوپروتئین ها هست، لیپوپروتئین لیپاز هستش، یا تنظیم عملکرد لیپاز رو انجام میده. بعد باز آپوئی هم باز به همین ترتیب. پس بنابراین اینها دارای نقش ساختاری متفاوت و نقش عملکردی متفاوت هستند. خب من این قسمت توضیحات قسمت ساختار و عملکردشون رو میذارم برای جلسه آینده خدمتتون توضیح میدم. بعد و اینکه اینها چه سرنوشتی پیدا میکنن توی بافت‌های مختلف. خسته نباشی درس میکنم خدمت همه شما. حضور همگیتونم وارد شد. من حالا البته فقط توی لیست آقای آرمان زاهدی، آقای رائد عبدزید داخل بنی سالم و آقای بهنام خراسانی رو اسمشون